Hello everyone, I made this recording to provide you with feedback on the last reassessment that we took. So let's get right to it. So question one, I did tell you before you took it that we were going to focus on the lactate dehydrogenase reaction. So in order to earn back points for question one, you were expected to write the structures and of the reactants and the products for this reaction. So if we look at pyruvate, if we look at in blue right here, that's the only part of the molecule that changes. The carboxylate group and the methyl group remain unchanged. But this is a redox reaction where we're going to donate a red proton and a blue proton to this carbonyl group on the pyruvate to form an alcohol group. And we need 2 electrons as well. That might not be as obvious, but we will need electrons as well as we go from NADH to NAD+. So this is the pyruvate is reduced to lactate, and the NADH, which donates the protons and electrons, is oxidized. So it's a redox reaction, and LDH is the enzyme that catalyzes that. So this is maybe fairly recent report that I read. So a group of investigators were looking at LDHA and LDHB isoforms to see if they can look at inhibitors that might inhibit one and not the other. So this is what they call the AXKO-0046. I'm not sure why they named it that, but anyway, that's the name of it. And we can see that LDHA, which is shown here in red, this is the concentrations of the inhibitor in molarity. This is on an exponent scale. So this is 10 to the minus 11th molar. This is 10 to the minus 4th molar. So that means that each one of these units is a 10-fold increase in concentration. So they looked at a very wide range of concentrations. So at the low concentrations, we don't see any inhibition. But when you get to, say, 10 to the minus 8th molar, we can see we're starting to see some selective inhibition of the LDHB. And definitely by the time we get to the 10 to the minus 4th molar, we're almost at 100% of LDHB being inhibited, where we haven't even reached 20% of inhibition for LDHA. So in your answer, I would have wanted you to say that you did notice that the inhibitor wasn't as effective at inhibiting LDHA. And again, notice that you don't even get 20% inhibition, even at high levels of the inhibitor, which is 10 to the minus 4th molar. I did expect you to express this properly. I'm not sure why they expressed the graph this way, but I needed some indication from you that you knew what these numbers meant. Okay, so to continue, the LDHB is inhibited at a concentration as low as 10 to the minus 8th molar, and then we get this nice dose response as we increase the concentration of the inhibitor from 10 to the minus 11th molar to 10 to the minus 4th molar. So when we get to 10 to the minus 4th molar, we're nearly 100% inhibited for the LDHB. So we have some selectivity here. So because the inhibitor inhibits LDHB and not LDHA at low concentrations, we do have the potential to use this therapeutically as a drug. However, in the background, we have to note that some cancers have upregulation of LDHB and some LDHA. So the colon cancer and the triple-negative breast cancer have upregulated values of LDHB. So for those, this inhibitor would be possibly effective, but not LDHA because that isn't upregulated in those types of cancers, and in fact that's upregulated in other types of cancers. But you need a different inhibitor for that, and those do exist. But also in the background, you know that LDHB is also found in the heart. So if you did use this therapeutically, you'd have to keep this in mind and monitor their heart health of your patients. Okay, so question 3, I gave you this mechanism. Again, I just sort of gave you a snapshot. So here we are in the active site, and in blue, this is pyruvate. Now, they just showed the R group. The R group is the methyl group, and I have the pyruvate and the lactate just so you can see where's the substrate, where are we starting and where are we going. So the methyl group doesn't change. This carboxylate group doesn't change. The only thing that changes is this carbon number 2, where we have a carbonyl group here, and we accept 2 protons and 2 electrons to form the alcohol. So that's the mechanism, and we can see just a part of the mechanism right here. We can see the involvement of His 195, and this is the NADH, and we can see that the NADH is shown donating its first proton, and we're going to get a second proton from the histidine. There are more steps, but this is all I showed you. We do see that we have aspartate and arginine, another arginine over here, and there's also either an arg or a glin, depending on which species of LDH you're looking at. But I didn't draw or the artist didn't draw the structure of that, and I didn't ask you about that in the question. So again, think about where you are and where you're going. So the histidine right here, so this is histidine 195. So we can see that we have sort of this concerted kind of proton donation going. So the histidine has a proton, and that proton is going to be donated to this oxygen, and we're going to form a bond between the oxygen and the hydrogen, and that's going to form our alcohol group. So what this means is that we don't know what the pKr group for free histidine would be about 6. So the histidine 195 has to be probably greater than that, because even at physiological pH, that would be maybe 90% protonated, but we don't know, but it has to be higher than that. The point is we have to have a histidine 195 that has a high proton binding affinity. It has to have a proton, because if it didn't, we wouldn't be able to donate it. So there's our answer right here. So the His 195 is initially protonated. So it has to have a higher pKa, because remember, high pKa is high proton binding affinity. We note that it acts as a general acid catalyst. Acids donate protons, and we can see that this proton is being donated to this carbonyl group, which is going to help form this OH group. And again, we can see that that's shown here in blue. What about ARG 171? Well, again, if I look at where I am and where I'm going, that carboxylate group does not do anything at all. So if you're looking at arginine 171, this functional group is not going to change. So what is likely then is that if I have an arginine which is positive, this is a guanidino group, it's resonance stabilized, which is sort of drawn this way. It's, see, I have like a bond and a half and a delocalized positive charge. That guanidino group is resonance stabilized. But we can form a nice ion pair here between the arginine and the carboxylate group of pyruvate. So what is the arginine doing? Well, it is probably positioning the pyruvate in the active site properly, because you have to do this properly so that this proton donation can happen. We're stabilizing the binding of the pyruvate, but since this is a bit of an intermediate we're showing here, we will likely be stabilizing the transition state as well, because the arginine is right here and we've got this negative charge, and the arginine is in a perfect place to do that. All enzymes stabilize transition states. So if you're ever asked a question like this, that's got to be in your answer somewhere, because that is how the enzymes decrease the activation energy and speed up the reactions, is by decreasing the energy of the transition state. So what about ASP 168? Well, over here again, so if the histidine is protonated, it's going to be positively charged, so we can form this ion pair right here. But the ASP must be doing something else besides that, because the His has to be in a position to be able to donate a proton. So there must be some kind of like a polarizing effect, that the ASP has to facilitate in some way this proton donation, because that otherwise the reaction is not going to happen if this proton doesn't get donated. So it's like the aspartate and the catalytic triad of chymotrypsin. So we've got an ion pair, we can stabilize interactions between neighbors, because histidine and aspartate this is 195, 168. They're far apart in sequence, but they're close together when you fold them up. But there has to be some way to get this thing going, and the ASP 168 does that. And in the last question, I gave you some data from the paper. So I plotted this on Excel. So this is the line without the inhibitor, and these solid lines, and the dashed lines are with the inhibitor. So we can see here that the this is an appearance of an uncompetitive inhibitor. So if I go back to the structure of this inhibitor, if I go way back here, we can see that this does not look at all like anything that can bind to the active site. And we can look here, and there's our active site. And we can see that if the pyruvate and the NADH, those two substrates, are binding to the active site, we can see that that inhibitor is not going to bind to the active site. If you read the rest of the paper, there's some evidence that that this inhibitor has something to do with interfering with forming higher order oligomers of this enzyme, dimers and then tetramers. So it's not binding to the active site; it's binding somewhere else, but it's somehow making it impossible for the substrate to be converted to product. And it could be, because we're working with an enzyme like this in Biochem Lab, that if the enzyme is a monomer, sometimes if you have enzymes that are dimers or tetramers, if they're monomers, they don't work. So if you can prevent the dimer or the tetramer from forming, then you can prevent the enzyme from carrying out its function. So I didn't have you go through all of that because most people did very well on the exam because you've had a lot of practice at this. But I did want to give some people another chance to look at the Ki. So again, you would have to identify this as uncompetitive. So this would be the Ki primed. Sorry, I misspoke there. You would need the Ki primed here. So because alpha is equal to 1, but alpha prime is greater than 1. And we can see that we are going to decrease both Vmax and Km by the same amount. That's why if you look at the slopes are the same because you actually have an alpha primed here and an alpha primed here and they cancel out. I thought the easiest way to figure out Ki primed was to look at this table and then realize that first I had to calculate the Vmax without the inhibitor. So again, if I look at without the inhibitor, that's 0.14 reciprocal, so it's minutes per micromolar. Then I get micromoles per minute as my Vmax. And then again, if I know that the y-intercept is going to change by this factor of alpha primed, then what I do is I just cross multiply here. So Vmax times the y-intercept is going to be equal to alpha primed. So that means if I take the Vmax without the inhibitor, multiply that by the y-intercept with the inhibitor, then I can see that I increase my y-intercept by 4.8. So that's the value of alpha primed. So then I just sort of substitute in, and that's my concentration of the inhibitor that was giving it to me. So my Ki primed is 2.6 micromolar. Another way you could have done this, I think this is more work, but this would work, is you calculate both of the Vmaxes, both without the inhibitor and with the inhibitor, just by taking the y-intercepts of those two. So then what you could do is you could just compare them because the Vmax decreases by this factor, and you get 4.8 there too. So you would get the same answer. The other thing you could do, although this would be a lot more work, is to calculate the Kms, but then you'd have to use the Km, you'd have to calculate the x-intercept, and then you'd have to take the negative reciprocal to do Km with and without the inhibitor. And again, if you take the ratio, then you should get 4.8. So that's a third way to do it. So that's sort of like I'm presenting do this in sort of like a difficulty of doing this. Anyway, so those were the 4 questions that I asked you to look at, and I really appreciated reading your reflections and looking at your discussion posts on Canvas. And if you have any questions, I'd be happy to talk to you in office hours and answer all your questions. But hopefully I've answered your questions here. Thank you very much for your time and attention, and I will see you in class.